All right, everyone. Welcome to our first lecture of this semester. Before I introduce our invited speaker, I'd like to make a couple of announcements. One announcement is that this joint venture between the Federal University of Rio de Janeiro and the University of São Paulo has developed into a more complex relationship. We included the Federal University of Uberlândia, the Programa de Pós-Graduação em Imunologia e Parasitologia Aplicadas, which is coordinated by Thiago Mineo, and also the regional board committee of the Brazilian Society of Immunology. Okay, so we spread our wings and hope with that we can have more, increase our discussion and reach more people from our community. Okay, so thank you very much. And it's a great pleasure for me to introduce the opening conference of this semester. Our invited guest is Dr. Helton da Costa Santiago, great friend from a long time. Helton holds a degree in medicine from the Federal University of Minas Gerais, where he also obtained his master and PhD in biochemistry and immunology. Later on, Helton was trained as a postdoctoral fellow at the George Washington University, where he focused his work on vaccine development, part of what he was going to speak to us today. But he also did a postdoc in NIH in Bethesda, where he studied allergies and helmintic infections. Actually, Helton is an expert in different areas of immunology, infectious disease, and also cancer and vaccines, as you will hear. Helton is currently an associate professor, also at the Department of Biochemistry and Immunology at the UFMG, and a member of the, gestor is the management committee or management board of the CT Vacinas, where he is also responsible for the clinical trials, the platform, at particularly the SpiN-Tec. Helton was affiliated and member of the Brazilian Academy of Science, and he was also visiting professor at the Johns Hopkins Bloomberg School of Public Health, and so many other things. I will cut it short and just thank Helton for accepting our invitation at the last minute as well. Okay, thank you very much, Helton.
Thank you, Gustavo. It is a great honor and a great pleasure to give this lecture today. And before we start, I would like to make a propaganda of the symposium we are promoting at UFMG, the symposium about controlled human infection models. It's a procedure that is very used in developed countries, and the developing countries are trying to implement these procedures to accelerate vaccine development. And we are holding the first Brazilian symposium on controlled human infection models at UFMG. It will be this week on Thursday and Friday. You can check the program in this website. We don't have tickets for in-person attendance anymore, but we still have a lot of room for online participation. So everyone interested in clinical trials and controlled human infection models are welcome to attend. So today I'm going to talk about the development of SpiN-Tec. SpiN-Tec is the first vaccine, the first Brazilian vaccine to be conceived in Brazil and to enter in clinical trial in Brazil. So it had all its development done in Brazil. It's not the first Brazilian vaccine to enter in clinical trials. We have some vaccines in the past that has reached clinical trials, but they were not developed with the same standard that the vaccines today are required. Also, we have some other vaccines that were invented in Brazil and entered in clinical trials, but the clinical trials were not performed in Brazil. So this is the first vaccine to do the whole process of development in Brazil. And it is teaching a lot about the transitioning from the basic science to applied science to Brazilian scientists. So I don't need to talk much about the pandemic of COVID-19. We just finished the pandemic, although now it is an endemic disease like everybody has. It became a flu syndrome from a seasonal syndrome. So we expect to have peaks of COVID-19 every year now during certain seasons. But it was very scary while the pandemic was developing, and everybody could follow its development. And here we have several waves of the infection, and this huge wave here in the end of 2001 and 2021 and beginning of 2022 is the Omicron wave. And the vaccine started with an ancestral strain of the SARS-CoV-2, and very soon it started appearing several variants of concern, which are listed here. The last one was Omicron, but even after the Omicron, we started having sub-variants of Omicron. And although this huge wave here was the first Omicron variant, we are still having these small waves that is now permanent in the public health because of the emergence of subvariant. So the interesting thing is that each variant will make the vaccine developed for a previous variant obsolete. So as the virus changes, and the main protein it changes is the spike protein, which is this blue proteins here on the surface of the virus. The vaccines that were majority developed against the spike protein will lose its effect totally or partially. And because of that, there is an interest in the literature to find new vaccine targets. So there is a run now for conserved non-spike antigens to develop vaccines. And here we can see some of the papers touching this idea, and we can see that nucleocapsid or N protein is becoming an interesting candidate. And we have data to show that the T cell specific for nucleocapsid that produces interferon gamma presents a negative correlation with the amount of viruses in the upper respiratory tract. So this is true for T cells, combined CD4 and CD8, but also it holds on for CD4 and CD8 T cells. It means that as more T cells responding to N protein, you have a better control of the virus in the upper respiratory tract. And CT vacinas, when the pandemic was developing, everybody in Brazil was running to find a vaccine for it. I mean, everybody in the world was trying to develop vaccines against the virus. And CT vacinas was also developing vaccines, and we had several candidates. And one of the candidates that really showed some promising was this SpiN protein. SpiN was a combination of the N protein, which changes way less, and is responsible for majority of the T cell responses, and the RBD portion of the SpiN, of the spike protein. So it's a chimeric protein combining these two regions, and that's a difference from most of the vaccines that were approved and used during the pandemic. Most of the vaccines used the whole spike protein, and what we see is that as new variants emerge, these vaccines start losing their efficacy. And while the SpiN-Tec focused on N protein, we, at least in animals, we are seeing the similar level of protection with different variants that we test. I'm going to show this data right now. But this was the work that was done by Julia Castro, and she used the SpiN protein to stimulate cells from individuals that were vaccinated with coronavac or that was convalescent of COVID-19, and both produced lots of interferon gamma when stimulated with the SpiN or with N protein or RBD. So she vaccinated animals, mouse that express the human receptor for the virus, with the SpiN on day 0, and then 21 days she gave a boost and waited for 30 days and then challenged the animals with SARS-CoV-2. The vaccinated animals, they produce interferon gamma either against RBD and against N protein. This interferon gamma is being produced by T central memory cells and T effector memory cells. And the animals survived. Here we can see a graph where Julia tested the mechanism of protection. Here is the weight of the animals. When they are infected, they usually lose weight, as we can see here. And the animals vaccinated, they preserve their weight. And here we can see the mortality. Infected animals, they die between day 7 and day 9. And she vaccinated the animals and she challenged the animals, but before challenge, she depleted the animals for CD4 here in blue. And there is a partial loss of protection. She challenged the animals. She depleted CD8. Also, she observed a partial loss of protection. But when she depleted both CD4 and CD8, the animals died completely, showing that the animals vaccinated, they need T cells for protection. So to test if there is also a protection associated with the serum, it means antibody, she transferred the serum of mice non-vaccinated that just received the adjuvant, and they did not protect against the infection. She transferred the serum of vaccinated animals to non-vaccinated animals and then challenged them, and they also died, all died until the 9th day of infection. So and here is the vaccinated animals, they don't die. So the protection was not being transferred by serum. It means that antibody was not playing a major role in this protection, but the T cells. She did the same experiment with the AstraZeneca and the animals when depleted for CD4 and CD8 and vaccinated with AstraZeneca, they don't die. I mean, they were vaccinated, depleted CD4 and CD8, and challenged, they don't die. So the participation, it shows that the participation of the cellular immune responses is minor for AstraZeneca, and the protection could be transferred with the serum. Here is animals that were non-vaccinated and received serum from naive animals. They died until the day 9, but when they received serum from animals vaccinated with AstraZeneca, they survived, showing that the antibodies was protecting these animals. So we have two different vaccines here. One that is stimulated T cells, and the protection mechanism is associated with T cells, and another one, and this is the mechanism of the majority of the vaccines that were developed, they have their protection mostly associated with neutralizing antibodies. When we challenge the animals vaccinated with the SpiN-Tec with Delta, they also survive. The Omicron is less virulent than Delta, and they don't die, they don't kill the animals. But the vaccination with the SpiN-Tec reduced the virus load in the lungs and protected the lungs from pathology. Here we can see the animals vaccinated with adjuvant only and challenged with Omicron, and we can see a huge infiltration with areas of hemorrhage. And of course, you look at these lungs, you can conclude that the gas exchange is compromised in these lungs. While when you look at the vaccinated animals, the lung is fairly preserved. There is an inflammation. We can see important inflammation, especially peribronchial inflammation. But the surface of gas exchange, the alveoli of the lungs are fairly preserved, allowing the gas exchange in the lungs. So show that SpiN-Tec was protecting in similar manners with Wuhan strain, with Delta strain, and with Omicron strain. But when we reach the point that we could think about the clinical trials, and just to be clear, I didn't participate in this previous results because I got involved with the SpiN-Tec development when they were looking for someone to develop the clinical trials. But when we reached this point, there was vaccination in Brazil already. And we took some time to reach this point. I'm going to explain why. But when we were ready to start our clinical trials, the population of Brazil was already receiving Coronavac or AstraZeneca and the Pfizer was starting already. So it didn't make any sense anymore to develop a vaccine to vaccinate naive individuals. So we decided to change the strategy to develop a vaccine that would be a booster vaccine and provide the other vaccines that are more associated with antibody mechanisms with what it is missing, like the T cell stimulation. So what we did, we in animals, we vaccinated animals with coronavac or SpiN. The SpiN-Tec per se does not induce neutralizing antibodies, as we can see here. But when SpiN is used as a booster for coronavac, it gives even a better booster for neutralizing antibodies than coronavac itself. And the same is true for AstraZeneca. When the animals receive AstraZeneca as a primary vaccination and a boost with the SpiN, it induces a little more antibody that makes it different from just the primary vaccination. So this data showed that SpiN-Tec seems a good booster vaccine. So we decided to go on with the clinical development of the SpiN-Tec as a booster vaccine. And here comes some of the challenges we faced during this development. For example, we are all basic researchers. The basic research in Brazil is very solid, very robust. We have very good basic scientists. Also, we have very good clinical scientists in Brazil, but we don't have in Brazil the people who do the transition. And now SpiN-Tec was facing this transition. And when you go to Anvisa and say, look, I have this awesome vaccine that we would like to perform clinical trial. First thing Anvisa will ask you is about GMP production of this vaccine. And then usually basic researchers vaccine was like GMP what? So GMP is good manufacturing practice. It's a form of doing the experiment that usually we don't care very much in basic science, but it's very important to prove the experiments and to show the evidence of the quality of the experiments to regulatory agencies. So first thing, we need to develop the vaccine under GMP production. And this is a whole new technology. Also, we needed a technology of purification for this vaccine to meet the standard for human use. And that's all a different level of purity that the vaccine must reach. And also we need a study of protein structure and quality of this protein, which is also something that usually we don't do when testing the vaccine in animals, in basic science. So Natalia, she took the challenge and she developed this technology with the standard that Anvisa would request. And then we could have a very pure, very characterized protein that was done under GMP conditions. And then we were ready, we thought we were ready to go. And then we invited Graziella to fill the documentation for Anvisa. And then Graziella, she's a pharmacist, and she was from industry. And when she came, the first question was about the formulation and which adjuvant we're using. And we were like, oh, we are using these adjuvants here in the animal testing. And she was like, this is not enough. You need a validated adjuvant. You need adjuvant produced under the right conditions. So, and you need the formulation of the vaccine well characterized to start the ANVISA application. So she developed the actual adjuvant we use. She found that one of the adjuvants we were thinking to use was with the patent expired. So she made a similar adjuvant at CT Vacinas, and developed the quality of GMP production for this adjuvant, and she transferred this for a company. Cristalia is a Brazilian pharmaceutical company that is a partner in the development of SpiN-Tec, at least in these first steps. Cristalia was a great partner. And she developed the formulation of the vaccine, and she developed and helped to perform all the testing that ANVISA would require for application, like stability, specificity, putting the vaccine in vials. And after that, we needed to repeat all the animal study with the final product. And this animal study should be performed under good laboratory practice. So a new level of standard for the animal study. So this was done by Natalia. And she developed the GLP test for immunogenicity that are really rigorous to meet the standard of ANVISA. And she also developed the animals for efficacy and protection to meet ANVISA standard. And she also coordinated with the CMP the test of toxicity. And we could meet all the requirements for the ANVISA application. So with that, this document was filled, Dossiê de Desenvolvimento Clínico de Medicamentos, the Brazilian equivalent of IND of the FDA. It's a very robust document. It can reach over 3,000 pages of documents and evidences of the candidate product. Also, we had to prepare an investigator brochure where you summarize all the development of the vaccine until that point, and to prepare the DEC, which is the study protocol. With that, we could have the approval of CEP, CONEP, and ANVISA to start. And you can see that we have quality control all over. All these steps, all these milestones needed to be well controlled in terms of quality. And we have Patricia Pito, that is the responsible of the quality control of CT Vacinas. She just does that. She goes to all the documentation. Of course, she doesn't do that alone. She has a team. She goes through all the documentation to guarantee that everything is under good annotation practice. Everything is under a SOP. Everything is the forms has been filled correctly. So she's responsible for keeping the quality control of all these steps. And ANVISA may just come at any moment to do a audit. I forgot the term in English, but they can come to check all the documentation, and Patricia is the person responsible that if ANVISA comes, everything is in place and the study is approved. So here is the timeline of the vaccine development. It was conceived in April of 2020. In July, we had the protein expressed, and in November, we started the experiment in animals. But just in September of 21, we had our first clinical lot. And this clinical lot is not the clinical lot that we use in the patients. You need at least two, because you need one to do stability test and determine a minimum stability of your product to be applied in the second lot. So we had a second lot in October, and then we repeated the toxicity studies and the preclinical studies with the clinical lots until February, and we could have approval of the ethics in July of 2022. In October, we got approval from Anvisa, and in November, we started the clinical trial. The clinical trial had this very complex title, which is a technical title that means clinical trial of first in human, phase 1/2, double-blind, randomized, controlled with active comparator to evaluate safety and immunogenicity of the chimeric vaccines SpiN-Tec, MCTI, UFMG, against COVID-19. The inclusion criteria was participants between 18 and 54 years. We were planning to do above 55 too, but we couldn't find individuals for that that filled the inclusion criteria, which also included individuals that needed to be vaccinated with CoronaVac as primary vaccine and have received one boost with Pfizer vaccine without previous history of COVID-19. For phase 2, we could be a little more flexible. So individuals of 18 to 85 years old, it's wrong here, it's 85 years old, and they could have received primary vaccination with CoronaVac and Covishield, and received boosters with Pfizer or AstraZeneca at least 6 months before randomization in our study. Also, they could have had COVID-19 at least 6 months before study participation. Here is the study design. We had 3 arms of the study, one receiving 20 micrograms of SpiN-Tec, one receiving 60 micrograms, and another one receiving 100 micrograms. Each arm had 12 individuals, 3 receiving AstraZeneca, and 9 receiving SpiN-Tec. And we have the first three participants as the Sentinel group. This is the first individuals to receive a vaccine in that dose. And these individuals were vaccinated, and we just could continue that arm after 1 week of observation of these 3 individuals. So so we vaccinated the 3 individuals, we waited 1 week, we evaluated the safety data, and if everything was okay, the other 9 individuals were allowed to enter the study, so they were vaccinated too. And after 2 weeks of these individuals' observation, we evaluated the safety data again and authorized the next arm. So we did that for all the arms. In the last arm, we summarized all the data and submitted to DSMB to approve the phase 1. And after that, we send the data to Anvisa. For the phase 2, we are using SpiN-Tec and comparing to Pfizer. And our plans were to enroll 180 individuals in each group, in each arm. However, we could reach the necessary sample size with less. So we vaccinated about 160 individuals in each group. And the individuals, they had a period of triage where we evaluated, we performed medical evaluation with a physical exam, history, lab work, routine lab work to guarantee they were healthy. Then on the day 0, they received the vaccination, and then they returned for visits on 7, 14, 28 days, and then 90 days, 180, 270 days, and 360 days. Our primary endpoint was 28 days. So after all individuals complete 28 days, we break the blinding and we make the calculations and evaluate. But the individuals, the participants, they keep being blinded, and also the centers that are evaluating them were kept blinded until the day 270. So during the triage and inclusion, the individuals had to sign the informed consent form. We evaluated inclusion and exclusion criteria. They were subjected to physical exam, hemogram, renal, liver work, infections, and other studies. At the day of vaccination, everybody was tested for COVID-19, and women was tested for pregnancy. They were vaccinated, and then they were observed for 60 minutes if they were sentinel group and 30 minutes all the other groups. And the individuals, they should fill a participant diary where they would fill and report all parts of things they were feeling in that day, everything that would happen to them, related or not to the vaccination. For example, they might have, I don't know, a bruise in their arm because they hit a wall. They would report that, and then under evaluation, we would see that and say, well, if he hit a wall, he had this bruise. So we would note bruise, non-related to the vaccine. But if he complained of headache, for example, and say, oh, I had this headache and the day after the vaccination, so probably was associated with the vaccine, and we would associate that with the vaccine. So in addition, the individuals would have phone calls on day 1, 3, 10, 21, and then monthly until the end of the study, which is 1 year, and presential visits on day 7, 14, 28, 19, 180, 270, and 360. So we advertised the study in the social media. We had this banner that was fixed in public places. The UFMG made some press release, and news outlets like radio and TV, they got interested. They covered the study, and we had a website for people to enroll, and here is what it looks like. So they start signing a form of consent, and then they fill some data. And we had a total of 1,700, almost 60 individuals that enrolled. And we vaccinated only 36 of them. The others were invited to the phase 2, but lots of them couldn't participate because they had received other vaccines or they had some background disease that would impair their participation. And here is the participant kit. So they received this bag containing thermometer, a ruler, so they could measure the size of edema or the size of erythema, the participant diary. If they had COVID, they would receive another bag containing also a saturimeter. And here is the big day where our first participant was vaccinated. It was a big party here at UFMG. We had the presence of the Minister of Science and Technology. This is the Secretary of Science and Technology giving the first vaccine. The president of UFMG is here, the State Secretary of Health, me, Ana Paula, Dr. Jorge Pinto, which is the PI of the study at the faculty at the School of Medicine. And here I would like to show you that our first arm, we started vaccinated in November. Gustavo, if my time is over, please let me know.
No, you have 15 minutes. That's fine.
Awesome. So we vaccinated our first individual in November, the end of November. And we've finished the third arm in May, but between November and January, we had another wave of COVID in Belo Horizonte. And between December and May, we had 5 cases of COVID. This is over 10% of our cohort. It's a lot of individuals. I think it also impacted even individuals that didn't report COVID because, as you're going to see, we had some variations in the level of some immunological parameters that could be because of this wave. So the 3 arms were broken down in 4 groups, a group of 20, 60, and 100 micrograms and AstraZeneca group. Here is the demographics of our population. Each group is very small, just 9 individuals, and it's very likely that we have some bias in such a small group. For example, the Covishield group, the AstraZeneca, they had more males, while SpiN-Tec of 20 and 60 had more females. But that didn't impact very much the analysis, at least so far. We don't have power for major statistics. And the main objective of the phase 1 is to guarantee that the vaccine was safe to move to the phase 2. And it was proven so. Here is the level of some of the parameters we analyzed during screening, and everything is very normal. These are very healthy individuals with no background condition, so not major stuff here too. And here is the general adverse events, like the number of things that the individuals complained. And we can see that we have the grading of association, like certain, probable, possible, and here improbable and definitely non-related. And if we just get the associated adverse events, the ones that we consider certain, probable, or possible, we can see that AstraZeneca had more adverse events when compared to SpiN-Tec in general. But because the group is very small, we cannot guarantee that SpiN-Tec is safer than Covishield. We can only admit that SpiN-Tec seems as safe as Covishield or safer. But regarding if it is safer, we are going to have the answer now in the phase 2, where we have statistical power for this kind of conclusions. But in general, we see that we could predict the adverse events, like over 80% of the adverse event was what we expecting them to be. We had, most of them were mild and moderate, and just one was labeled as intense, but this was a appendicitis, which turned out not to be related. And here is the most common adverse events. First is pain in the injection site, headache, tiredness, diarrhea, myalgia, nausea, arthralgia, and other adverse events which were less common. Regarding to the immunogenicity, we can see that Covishield, the AstraZeneca, could boost a little bit of neutralizing antibodies from day 0 to day 28, although it's not statistically significant. But remember, the group is very small for major conclusions on the immunogenicity. But we can see there is an increase in the levels of neutralizing antibodies in the AstraZeneca, also in the SpiN-Tec of 20, 60, and less pronounced in SpiN-Tec of 100. The same thing is true for neutralizing antibodies against Omicron strain, which also shows some increase with Covishield and AstraZeneca, and not so much with SpiN-Tec of 100. But notice here that this increase in Covishield, it jumps from 172 to 693, while the SpiN-Tec of 60, it started already from a higher background, a higher point. So we couldn't see much of this increment along the days, but it reached a level of neutralizing antibody even higher than the Covishield. And this background noise, I guess, is because of the Omicron wave we were having in when we started the study. We also tested the neutralizing antibodies against newer subvariants of Omicron, and we can see the same thing from day 0 to 28. There is an increment for all the vaccines, and none of them are different statistically from each other. When we have analyzed binding antibodies against the vaccine, Covishield didn't induce much of antibodies against the SpiN protein, and SpiN-Tec, it's good that it induced the antibody against itself. It's at the least we could expect. But anti-spike, we can see that the Covishield induced a little bit. The SpiN-Tec of 20 micrograms didn't do much, neither SpiN-Tec of 100 of 60, little bit for 100. But against nucleocapsid, the AstraZeneca doesn't do much, but the SpiN-Tec is more robust to induce the antibodies against nucleocapsid. Regarding to the production of interferon, here is the background without stimulation. And you can see there is this variance again in the background levels from the 0 to 14 to 28, probably associated to the Omicron levels, to the Omicron wave we were experiencing at the moment. But if we take a look in the levels here, it goes up to 250 micrograms, very few reaching above, levels above that. When we stimulate it with the SpiN, it jumps to almost 16,000 picograms of interferon gamma. So, and it's similar all over. So it's all the individuals had a very robust production of interferon gamma until now. But the individuals vaccinated with the SpiN-Tec could produce a little more against nucleocapsid, which is expected. When we look at where this interferon gamma is coming from, it is especially in the effector memory compartment and a little bit in the central memory compartment, exactly the place we expected these cells to be. So it is clear that we need more vaccines for SARS-CoV-2 because of the emergence of new variants. And our preclinical studies suggest that SpiN-Tec is able to protect against these variants of SARS-CoV-2 and with a cellular protection mechanism. The SpiN-Tec was found safe with a safety profile similar or even better than the AstraZeneca. And also it showed to be immunogenic, inducing neutralizing antibodies and interferon gamma against Wuhan and Omicron in levels that were similar or even better than the AstraZeneca induces. So our clinical data and our experimental data too, they suggest that SpiN-Tec is able to induce neutralizing antibodies against Omicron and possibly is a pan-SARS-CoV-2 vaccine. So this is the team that is helping all this process. We have collaborators from UFMG itself, from the School of Medicine of Ribeirão Preto, USP, George Washington University, and Fiocruz. And I would like to thank especially the, how did you say, Gustavo, the managing board of CT Vacinas. Ricardo, Ana Paula, Santuza, and Renan. Our sponsors, which is very important. And this, I'm going to jump and invite you again for the Brazilian Symposium on Controlled Human Infection Models that we are hosting at UFMG this week on Thursday and Friday. Thank you so much for the opportunity to show the results of SpiN-Tec. We are learning with this vaccine the bottlenecks of vaccine development in Brazil, and this vaccine helping us to solve these bottlenecks and advance the development of vaccines that is necessary for Brazilian population. Thank you so much, Gustavo.
Thank you, Helton. Super thanks. Wonderful presentation. It was a great way to start our semester, not only with science data, but also with hope that one day we will be very proficient in producing all kinds of vaccines here in Brazil. And really, you guys make us proud of not only, as you said in the beginning, of the Brazilian science basic, but also the way you built up, like a company, like, this initiative. It's just very impressive. And I don't know if people had this, particularly students, if they had this idea how complex is to go from an idea to the science, and then to break up all these, regulatory hindrances that you guys did so well. I think it's really a lesson to all of us. But anyways, let me see if somebody has questions. Normally we ask the students in either Rio, Uberlândia, or São Paulo. I guess they are a bit shy. It's tough. First day.
But this is true, Gustavo. You mentioned that we needed something more different from the academia. And CT Vacinas is really something apart. It's a mix of academia and industry, because it does this transition between academia and industry. And the level of regulatory tasks that we must accomplish to do this transitioning is really complicated. The slides that I jumped, let me go back a
It's
little
incredible. I had the opportunity to visit, as you know, and I was really impressed with the quality control of each individual step that you, from the production of the reagents to the way that you perform your experiments. It just makes us look like amateurs.
It's not. It's what it takes to do the transition. If we do what we do in the CT Vacinas, in your lab, we would kill the innovation. Because you need some freedom to perform experiments, to try out, when you develop your product, your vaccine, your medication, and you want to do the transition, then during this transition, you cannot be smart anymore. I mean, you need to be smart in a different way. Yeah. When I went to George Washington University to be trained in this, Jeff, he told me something that is very important. He said to me, here we do very simple stuff in a very sophisticated way. And that's true. You cannot do complicated experiments anymore, but you need to do simple experiments in a very sophisticated. And this is what kills the innovation. We do our experiments, we invent stuff, we create stuff in the basic science, but that to be put in the arm of someone and start the clinical testing needs a whole different level of development, which someone that has the mentality of basic science look and says, look, this is very complicated. I don't want to do that. It's very common to have this. Someone got to do that. I hope the industry does that.
I'm happy that you guys are doing that.
Yeah. So, but the industry looks at that stuff and says, no, this is very preliminary results. They are nice, but it's just a promise to invest so much money in such a risky idea. So, and that kills the innovation. So you need someone that is willing to do this transition, to take the risk. And Cetevacin is doing this, at least for vaccines in Brazil.
Yeah. All right, excellent. So we have a question from Lucas. Lucas, can you open up your mic and ask the question, please? Nope, I can't hear you. It's mute.
Lucas, we cannot hear you.
Yeah. So I think that while we were waiting,
It's
Lucas...
coming to my computer.
Okay,
question.
Okay. All right. Otherwise, there's Isabelle and Jan, the other people can just jump in. Raise your hand, open up the mic, and ask the question, please.
Okay, I think you guys can hear me now.
Yes.
Right? Okay, so thank you, Helton, for your talk. It's always very interesting to get a different perspective on science. I have a question, and I think I don't think that it matters that much because of course we have a great vaccinal coverage here in Brazil, but more for a conceptual standpoint. Do you think, what do you think is more important when you consider the type of response that you're establishing, that you want to establish with your vaccine? Like, if you have first a great humoral response with antibody production, and then a later cellular response with memory T cells, or do you think do you think it matters more to have first the antibody production and then the cellular response, or do you think that the order doesn't matter? So if you're thinking of developing a vaccine for another virus that is also a respiratory virus, let's say in a few years that appears again, do you think if we would try to develop a new vaccine for this hepatitis virus, it would matter more to focus first on the antibody response and then maybe try to achieve the cellular response with a boost? Or do you think that the order itself doesn't matter?
Well, the ideal is to have both at the same time. But everybody that used to work with vaccine development for viruses, they love neutralizing antibodies. It has been for decades like this. And even for COVID vaccine, all the first vaccines, they were focused on neutralizing antibodies. When even the SpiN-Tec, when the first experiments came out and showed it didn't induce neutralizing antibodies, the people at Cetevacinas told Julia, oh, just give up of this protein. It's useless. It's not going anywhere. And Julia insisted, say, no, it is protecting. So she kept doing the experiments. End up that this vaccine stood up and it's reaching this level of development. But what COVID is teaching us, that it's easy for the viruses to change and evade the neutralizing antibodies. And we see that every year with the flu vaccine. You use a vaccine, you induce neutralizing antibodies. Next year, you need to vaccinate again because you lose the levels of neutralizing antibodies because the virus changed. And COVID is even worse than flu. HIV is even worse than COVID. So we see that at least for COVID-19, a vaccine that induces a cellular response will have a longer usage time, because the virus will change way less in the compartment of T cells. Well, the N protein changes way less than the spike. In addition to that, they need to attach to the MHC of the cells, which select even more conserved sequences. So it's been very stable. The data in animals show that the SpiN-Tec is protecting equally. It doesn't prevent the infection, but the infection is very mild. So, and very often a vaccine that just make the infection mild enough is okay. Like you just have very often asymptomatic infection. It doesn't change anything for the public health. If you have a sterilizing vaccine or a vaccine that causes the infection to be asymptomatic, the result for public health is the same. And we hope that regarding to a booster vaccine, SpiN-Tec is offering what the other vaccine doesn't offer. So we are very excited about that. But this is a great question, and I think we need both. Optimally, we need both.
Great. Guys, there are people that already submitted questions, but I'd like you guys to open the mic and read the question. Or if you don't feel like asking English, you can ask in Portuguese, doesn't matter. But I can see Douglas, Ali, Dan Anderson, Isabelle, Ian, Bárbara, Carolina, come on.
I have a question.
Okay, great.
Helton.
Just raise your hand, guys, so I can see the line.
Thank you for your presentation and your great work, because nowadays it's becoming more difficult to do basic research and translate that into a clinical trial and get that to be approved. And my lab actually works with dendritic cell based vaccine for tumors. And right now we are undergoing a transition between phase 2 and phase 3. And one of the problems we are facing is because we need a lot of, we basically need a big grant to do this transition to phase 3, because we don't have the money to recruit people and to pay for everything. So if you are already planning this transition for your trial, could you give like some advice for us so that we can try and make this approach and basically to try and approve the vaccine for public health?
Yeah, we are in the process of preparing our phase 3 now. And you're right, it's very expensive. It's very complex. The study per se becomes simpler because phase 1, it's very complex because of the safety. Phase 2 is complex because of the immunogenicity, but you can repeat that during phase 3, and you're going to repeat, but you don't need the level of sophistication for phase 1 and phase 2 now. You can do a simpler study. But the challenge now becomes, at least for vaccine, becomes logistics, because it becomes a very big study. To approve a vaccine in Anvisa, we need to vaccinate at least 3,000 people with the vaccine. This is the minimum. So for vaccine, it becomes very challenging. A efficacy study for a cancer study doesn't need to be that big. You can do an efficacy study with a smaller group, and it will be still a new health product. And I don't have the details about a cell therapy how many individuals Anvisa will request receiving a cell therapy to approve its use in the public health. For vaccine, I know, vaccine and medication, 3,000. But my suggestion would be to talk directly to Anvisa. They are very open. They have channels where you can interact with them and ask them what they expect, what you need. Of course, you need to read the RDCs, the resolutions of Anvisa very carefully and abide to them very carefully. But if you have doubts, you can come to Anvisa, schedule an audience, and ask them, and they will guide you. We did that a lot, and we keep doing that for the phase 3 preparation. We already had 2 meetings just to discuss how we're going to do the phase 3 and what they want us to do. And we showed them our plan, and they say, well, if you do this way, we're not going to accept this. So we changed our design, the study design, to abide to their suggestions. So it's talk to them. They are open. They are willing to help Brazilian science to advance. And congratulations, it's very nice that we have people moving, making this move in the area of cancer too. It's very important.
Thank you for your answers. It's going to be very helpful.
All right. We have time for a couple of questions more, guys. So anybody else? Anderson, go ahead. He's still there.
There is Isabella and Ian.
Gustavo, if you don't mind, leave the students to ask, because
That's
I
fine.
think it's
just
important.
that, okay, I see now.
I just want to say something to Helton. Patrão!
Yeah, Anderson is meu patrão. I'm here, man. At your orders.
Isabelle, go ahead.
Thank you for the incredible work and the lecture. And I understand that you use 3 challenge doses in this phase of the study, 20, 60, and 100 microgram, right? So what will be the challenge dose for the next phase, and how it will be chosen the dose?
Yeah, well, regarding to the safety, all the doses were similar. The dose of 60 had a less numbers of adverse events, but as I said, the group is very small, so we can have confidence. So we considered that all of them had similar safety levels. Regarding to immunogenicity, we didn't have statistically differences between the dose of 60 and 100. But we had some difference between 60 and 100 when comparing to AstraZeneca and the dose of 20. So 60 and 100 were similar doses also in immunogenicity. So we selected the smaller dose of these 2. So we did the phase 2 with 60, and this is also the dose that is going to move for the phase 3.
Thank you for the answer.
Jan?
Good afternoon. Can you listen me good?
Yeah.
Okay. Thank you for the presentation. Congratulations for the work. When I read in this year about protein, vaccine, I have curious about the absence process of production and the transport, like in adjuvant context. Because of this, I remember cost idea. So if you can speak a little about this, please.
You mean cost?
Yeah, eu vou tentar perguntar em
Pode
português,
falar em
certo?
português.
às vezes eu me embaralho um pouco nervoso. Quando eu estou estudando, eu estudo, né, a parte de proteínas, sabe? Aí eu fico um pouco curioso sobre o processo de rendimento, o processo de eficiência, quando eu estou falando da produção, que diz muito respeito ao custo, ou então quando eu vou pensar no transporte da molécula, até antes do tempo para manejo de administração, também fico em dúvida sobre o processo de estabilização. Que apesar dos resultados serem ótimos, o que é uma maravilha, em termos de aplicação prática, às vezes essas questões interferem muito, né? Eu fiquei um pouco curioso sobre isso. Gostaria que o senhor pudesse me falar um pouco sobre, por favor.
Sim, a gente tem feito 2 tipos de teste de estabilidade na nossa proteína, né? Na nossa vacina. Teste a 4 graus, teste a 37 graus. O teste a 4 graus, ela já foi aprovada para 1 ano e meio de estabilidade. Então, se você pegar a vacina e colocar em geladeira, ela aguenta 1 ano e meio. E a gente está validando em breve 2 anos de estabilidade. O teste a 37 graus é um teste de degradação acelerada, né? E você usa essa fórmula para estimar uma estabilidade a 37 graus também, aliás, a 4 graus também, né? Mas ela vale também para 37. Para 37, mesmo, a gente validou ela no momento para uso de bancada apenas, né? Que ela já está em 4 horas ou 6 horas, uma coisa assim. Você pode manter ela na bancada. Depois que você abre o frasco, ela pode ficar 4 a 6 horas em temperatura ambiente que ela aguenta, né? Sem perder as qualidades. Depois de 6 horas, começa a perder a qualidade. Com relação ao rendimento dela, e isso implica muito no custo da vacina, né? Ela teve que sofrer uma série de otimização para melhorar o rendimento, né? E isso implica também tanto na produção GMP quanto nos processos de purificação. Então, isso é feito um processo conjunto. Então, a Natália, que desenvolveu a produção da vacina, ela já desenvolveu o combo, expressão em alta quantidade e alto nível de purificação, porque você sai perdendo depois, né? Mas ela tem, a nossa produção, eu não vou saber te falar detalhes dos níveis, porque tem outra equipe que faz isso, então, assim, as equipes, né, são muito competentes nos seus ambientes, eu deixo elas trabalhar, entendeu? Talvez o Ricardo Gasnelli saiba todas as etapas, eu não sei, mas eu não acompanho algumas das etapas. Mas eu sei que a produção dela vai permitir que seja o nível de expressão e purificação, né, o rendimento da vacina, vai permitir que ela seja uma das mais baratas a chegar no mercado, e que é importante para a saúde pública, né? E o transporte pode ser feito a 4 graus, sem nenhuma perda da vacina.
Perfeito,
Okay,
obrigado.
guys. Anybody else? Otherwise, we will cut it short. It's almost 25 minutes ahead of time. No?
Gustavo, I think Merve has her hand up for a
Okay.
while as well.
Merv, go ahead, Merv.
Hello, Professor. Is there any differences in effect level of the vaccine between women and men?
That's a good question, Merv. I don't know yet, because our group in the phase 1 was too small. And I'm waiting for the data of the phase 2 where this question will be able to answer. But so far, we didn't treat in terms of sex separately. We just did all together. But that's a good question. I'm curious too.
Thank you so much.
Miriam?
Gustavo, if there is time for, I hope it's a quick question.
Go ahead.
Yeah, just one thing. Very congratulations on your talk. It's really neat to see it all together. And I wonder how easy would it be, or would it be possible to for the updates of the vaccines, like all the work that you have done and all the production that you have established as a group, is it easy to start add something else or start a new vaccine using the platform, or do you need to start it over again? I know that knowing the process is like 90% of the problem, but I wonder if you will be able to use that to update the vaccine or to use other specifics for that.
That's a great question, Miriam. And SpiN-Tec, as I said, is teaching us how to do it. We of course we are hoping that it makes to the pharmacy and to the arm of the Brazilian people. But if it doesn't, I think the best legacy this vaccine is giving us is exactly to open a road for development of new vaccines. So we learned lots of stuff. We learned all the processes which can be applied to other recombinant vaccines. So for this platform, it's very easy for us now to adapt what we have done at CT Vacinas to develop other vaccines. And we are already doing that. Our next vaccine to enter clinical trials might be a malaria vaccine that was developed by Irene Soares of USP. So we are finalizing the clinical development of her vaccine, and we expect to start the clinical trials in the first semester of 2025. Also, we have a vaccine for leishmania that is also entering in this pipeline using what we have laid for the SpiN-Tec. Now, CT Vacinas also have attenuated vaccines, virus recombinant vaccines, which are different platforms. But SpiN-Tec taught us a lot about what is quality control, how to do quality control, and the principles can be applied to even other platforms. So we are working on that too. So we hope that we'll be able to collaborate with many other groups in Brazil to get their vaccines and speed up the development, or to help them transition from the basic science to the clinical trials. Because of course, we have such a robust basic science in Brazil that I don't think CT Vacinas will be able to handle everything. But we are laying the road so that other universities can learn and copy from us and do their own product.
All right, guys. Thank you, Helton, so much. It was wonderful to have you here. And we are already, leave this invitation for you to come back with the news from the SpiN-Tec and perhaps others. So thank you very much. And everybody, see you around. Have a wonderful week, or we can meet again, rejoin next Monday at noon. Super thanks.
Thank you, Helton.
Again.